Hi, and welcome to this lecture on environmental adaptation of animals. On these last videos, we will consider on transportation of solutes and water, so this is still something that you should already be familiar with. And we will start with diffusion and facilitated diffusion. Let's think about hummingbird when it's drinking sugar nectars from the flowers. The sugar transportation is not so simple as it might look, that okay, it will just suck the nectar and then the sugar is available for the muscles, because we have cell membrane that is preventing the free access of all these solutes that are soluted in the water. And we also have the epithelia that is limiting the access for our bloodstream. So we will consider quite a lot on what happens on the membranes on these videos. If we take a typical animal cell and measure how much different ions there exist inside the cell and outside the cell, we can see that sodium, chloride and potassium are very typical, and then a little bit less on calcium, and even much lower concentration of protons, for example. And you can see also that some ions are more inside the cell and some are more common outside the cell. And two of these are very common in outside, but there is limited amount of these inside the cell, like sodium and calcium. So if there is a pathway on the membrane, sodium and calcium will flux inside the cell. Then we have potassium concentration difference. So potassium is very common inside the cell, but low concentration out. So if we have a pathway for potassium ion on the membrane, potassium is fluxing out from the cell. And then we have some that don't have so huge concentration differences, chloride and protons. So they can move in both directions. This ion distribution is essential for the homeostasis. So we have to be able to transport material like the glucose through the membranes. And this affects on both in the whole animal and also on the cellular level. We have a membrane that is forming an isolation layer, but so it's limiting the flow of potassium from the inside to outside, or sodium from outside to inside. But it also can be used for selective passive facilitated diffusion. So if there is a pore for sodium but not for potassium, then it's permeable for the sodium ions. But then we can also use it for transporting some other material together with these ions. But it also affects on the whole animal level. In freshwater fish, the pond or lake, there's a usually quite low concentration of ions, quite low concentration of salt in these waters. So there is a concentration difference. And so in the blood, there is much more sodium and chloride, and it will easily be fluxing away from the fish. So the blood would be too dilute. And at the same time, because there's more sodium and chloride in the blood plasma, okay, then by osmosis, the water would be accumulating in these fish. And also, when the hummingbird was drinking the glucose, we have the epithelia that is preventing the flux of all the material in the intestinal lumen to the blood capillaries. So we need to have some specific mechanism to take the glucose from the lumen and carry it through the epithelia all the way till blood capillaries. Well, the diffusion is limited with bowls. So if we have a cup of tea, we know that okay, the concentration differences tend to disappear. So if we wait a little bit, then we have the tea, it's the same color all around in your teacup. So that's why the particles in aquatic solutions, or when they are gas, they take all the space they can. And if we have a membrane that is permeant for particles, well, the flow will go toward the lower concentration, and these solutions are in chemical equilibrium, which means that, okay, in that case, the flow from left to right or right to left are happening all the time, but they are with the same rate. And if we think this in microscopic scale, we have a pore, and the pore can be occupied with these molecules or particles that are going through. And if we have a high concentration, it's more likely that these particles will find a pore in the membrane. And that's why they are going from the high concentration to low concentration. And this diffusion has been invented long time ago, and actually described in mathematical way 150 years ago. Alfred A. Fick invented in 1855 that, okay, solutes actually are behaving same way as flow of heat. He was not just studying animal physiology, he was studying steam engines. So the flux of these solutes depends on the speed of individual particles, and then it depends on what kind of material we have there around these particles. So if we have 4 walls, then the diffusion is smaller, etc. And this can be described easily that, okay, there is some kind of constant with the D that we have to take count, but then we have concentration C1 and C2. So the larger the concentration difference is, then more there is this flow of ions. And then it depends on X, meaning that, okay, what is the distance? And one extreme is the cell membrane. Cell membrane is very, very small. So means that, okay, the thickness is very small, and then the concentration difference can be quite high. And that makes it, okay, if you have a pore, the flux of ions will be very huge. But still, it doesn't work in the animal level. So in cell membrane, that is maybe 10 nanometers, the time to halve the concentration difference would take maybe 100 nanoseconds. But even in a small cell that is 10 micrometer, so it takes 100 milliseconds. And for cell life, that's a quite long time. And if we have a skeletal muscle where the length can be 1 millimeter, it takes already 17 minutes. And if we have a human eye lens that is about 2 millimeters, it takes 1 hour. Or the heart muscle that is about 2 centimeters thick, it takes 5 days. Or we have long nerves from the thumbs of our leg all the way till our pelvis. It takes 32 years to halve the concentration difference. So we can't use or rely on diffusion in our cells. Well, there are factors that are affecting on the diffusion rate. First of all, the temperature. So if we have a higher temperature, all the particles are moving faster. Then we have permeability. So if there is a pore for these particles, they can go, for example, across the membrane. Then they are moving faster if they don't so much interact with the water molecules. So this is something a little bit funny, because, okay, well, when we have particles that are soluted in the water, actually they are all the time interacting with water. And more water there exists around these, the slower they move. And then if the particle is also charged, it affects that, okay, it's not only moving because of the chemical gradient, but it's also affected by electrical charges. So if there's an electrical gradient in the solution, meaning that there's voltage difference, then it might be that it's affecting on the same way, on the same direction or in the opposite direction. So that's why if we think on an animal or a cells inside the cell there can be a huge concentration, and then if there is a pore, there is around the cell there is a little bit there is a huge difference in the concentration, but then by diffusion without the membrane far from the cell, there is much less. So it can be that, okay, it's accumulating also around the cells, especially these electrical charged particles. So we have electrodiffusion. So we have cell membrane, and depending on the voltage difference between inside and outside, it tends to accumulate positive and negative charges on this membrane. And that's why now if we have a pore that is permeable, permeant for ions, for example, then okay, we have a concentration difference from 150 to 50 millimole. So okay, we know that, okay, we can calculate, okay, how much the flux could be in these cases. But at the same time, the flux is also modulated by certain factors that are poor electricity. And this makes the equilibrium potential of ions something else than zero. So for an ion, for example, that is inside the cell there is 150 millimole and outside there is 5, then it depends that, okay, both on the concentration difference but also on the voltage of the membrane, that, okay, are the ions moving inside the cell or outside the cell. So these charged particles the direction is affected by both concentration and electrical gradients, and they can be in the same direction and on the different direction, and this causes that all the ions have specific equilibrium potential. So actually this means that, okay, when we have a membrane and now we have an ion that is moving, the concentration gradient and electrical gradient can be in the same direction, and then therefore the diffusion can be fast. But it can also be that the electrical gradient can be different, limiting the diffusion, because the concentration gradient is in one direction and the electrical gradient is in the opposite direction. And that's why we can have the equilibrium potential of different ions. So we have in a normal cell, there is a potassium inside, sodium outside, and etc. So the sodium is far from its chemical and electrical equilibrium. So it seeks all the time a road to flow inside the cells, and this would depolarize the membrane. But at the same time, in a typical cell, the potassium actually is quite near the electrochemical equilibrium. So it's if there is a pore, usually there is a pore for potassium in these conditions, and it can flux, but it doesn't flux so much because it's in the equilibrium. But if the sodium is finding a place where it can flow inside the cell to depolarize the membrane, then the potassium would flux out from the cell to inhibit that. So it would be repolarizing it. So it's inhibiting the change in the membrane voltage. And this electrical potential can be it can be affected by flow of ions, but then the electrical gradient is also affecting on the concentration gradients. So if we have two ions and two cations and two anions, and the membrane is soluble for one ion, so it will reach its electrochemical equilibrium. So when it's flowing, then the problem is that, okay, it will make a voltage difference on this membrane, and these accumulate the positively charged ions and cause that, okay, limiting the flow of more potassium from one side to another side. The other way that the thing that it's also limiting, it's that, okay, the electron neutrality in this bulk solution, it should be always 0. So this is quite often used for chloride transportation. That, okay, when you are moving chloride, then the sodium is or some other cation must go together, and this affects that, okay, now you have more salt inside the cell, and that will get water to go inside the cell, and that will swell the cell. So it's sometimes this chloride is getting quite complicated. So we have different kind of ion channels. We have ligand-gated channels, voltage-gated channels, and other channels. And as you can see, there are several members on each of these. On voltage-gated channels, we have channels that are limiting the pore, that, okay, only sodium can go through it. We have channels that are calcium selective, potassium selective, metal selective, proton selective. Then we have other channels. We have channels for water, so these aquaporins. Then we have channels for chloride. And then we have channels that are ligand-gated. They can be receptors of neurotransmitters, but they can be also other kind of ligands that are affecting that, okay, will the channel be opened or closed. And if we summarize that how these work, well, the voltage-gated channels are perhaps the most well-known, because they were the ones that were found in the 50s by Hodgkin and Huxley. So and these are quite often described in, for example, in the high school books. So in the protein, there are some positive charges in the transmembrane area. So somewhere between the cytoplasm and extracellular fluid. And if the membrane is changed, membrane potential is changing, then this charge tend to move either outward or inward. And actually this is the way how it affects on the gate that is opening or closing the channel. So when we have a membrane potential changed, then this pore will be open. And the regulation is very simple. So they form actually identical responses. For example, in neuro action potential, every action potential looks a little bit, quite much the same. Okay, then we have stretch activated channels. So some channels are glued in the cytoskeleton. And usually these channels are a little bit much less characterized than, for example, voltage-gated. but it can be used to detect that, okay, cell movement. So if the cytoskeleton is physically bending the channel, it will be opened. And this can be used for hearing or, for example, the regulation of epithelial cell division. So if there is you lose your neighbor cell, that affects on the tension of your cell membrane and affects that, okay, you start the cell cycle. Then we have ligand-gated channels that are affected by, for example, neurotransmitters. And that's very simple. So we have a ligand, either outside the cell or inside the cell, that is binding on its receptor, and then the receptor opens the pore and ions can go through. But it's not only limited on neurotransmitters or hormones, but also, for example, chili pepper receptors, etc. And then we have channels that can be affected by phosphorylation. So using the ATP to add a phosphate in the protein can affect that, okay, now it's more open. But actually, quite often they are voltage sensitive channels or ligand sensitive channels, and this phosphorylation is affecting on either on voltage sensitivity or ligand sensitivity, or that the behavior that, okay, when they open, they will be longer time open or less time open, etc. Thank you.